Hi, you guys. This is going to be part two of our protein synthesis lecture. So, this one is on translation. So, again, we already finished transcription, which was taking the DNA and transcribing it into mRNA. Now we're ready to take that mRNA and read it and turn it into a sequence of amino acids. M in mRNA stands for message. And now we have another molecule called tRNA that stands for transfer, and that's going to interpret the message at the ribosome. So the ribosome is the site of protein synthesis, in that it is where the amino acids get brought and put in order. tRNA is a funky looking shape. It is made in the nucleus from a sequence of DNA, right? So DNA is coding for how to make these tRNAs, and then they get released out into the cytoplasm where they actually go to work. They are specific for each kind of amino acid. So at the 3' end of a tRNA, that is where it is going to bind to an amino acid. On the opposite end, somewhere along its complex folds, is the anticodon, which is a sequence of 3 letters that complementary base pairs to the mRNA codon. And then we have this weird phenomenon known as wobble, which basically means that the third base doesn't really bond that closely. It's really the first 2 letters that bond, hydrogen bond to the codon. That third base is not as strict. So AAG and AAC could probably bind to the exact same codon, regardless if that last letter matches or not. But you're still going to bring the correct amino acid, because a lot of those codons code for the same amino acid anyway. So it's called third base wobble, and it's just another way to illustrate that multiple codons code for the same amino acid, and for good reason. This is just a cool word. Of course, there's an enzyme that does this. There is an enzyme that binds the amino acid to the tRNA, and it's got a fun name. You could try to say it: aminoacyl tRNA synthetase. Fun to say. And it's an enzyme that attaches the free-floating amino acids to the correct transfer RNA, and it uses energy to do so. And then we call these tRNAs that have amino acids on them, we call them charged. I need my other hand. Charged. So a charged tRNA is one that is locked and loaded with an amino acid. Once charged, it goes to the ribosome, and that's where it will meet up with the messenger RNA. The ribosome itself is also an RNA complex. It's RNA wrapped around some regulatory proteins. These are made in a small area of the nucleus called the nucleolus. Again, these sequences of our RNA are coded for by the genes in your DNA, but then they get assembled with the proteins in that structure, and they pop out of the nuclear pores and onto the rough ER, or they pop out into the cytoplasm. And they are generally existing in 2 subunits, in 2 separate parts that only combine forces when an mRNA binds to it. Ribosomes have their own active sites, their own binding sites for the tRNAs. They have an A site, which again, I think stands for, it stands for this enzyme that's got a huge name, aminoacyl tRNA. Binding site, I think of it as arrival. So I think A for arrive, P for park. Not necessarily worrying about this enzyme name. I just think of it being parked. Here in the lecture, it says P stands for polypeptide, because that is where the polypeptide chain is formed, or where the peptide bonds are made. And then you have the exit site, E for exit. So initiation involves mRNA binding to the smaller subunit. The large subunit gets called over and attaches itself, and then we can begin elongation. The presence of that tRNA carrying methionine always carrying methionine because AUG codes for methionine. Every single time that arrives at a small subunit, that signals the large subunit to come over and bind. And that's what allows the other ones to have a spot to bind. Because now that the large subunit is there, now there is an arrival site. Now there is a binding site for more transfer RNAs. So the elongation part is exactly as it sounds. It starts to make the protein longer. So you're going to get a tRNA carrying another amino acid shows up at the arrival site. This is going to be read 5' to 3'. It adds at that 3' end, or those 3 codons. It donates its amino acid to the polypeptide chain. Again, as soon as those amino acids get next to each other, there is that catalytic portion of the ribosome that initiates that peptide bond. It's going to remove a water between the carboxyl of one amino acid and the amino of the one next door, and that's what creates the peptide bond. Then we get a little bit of movement or shifting. So then it's called translocation. The ribosome moves or shifts down another set of 3 that pushes the tRNA to the E site or exit. It's dropped off its amino acid, so it can get the heck out of there. And then a new one will arrive. So we just keep doing this over and over again until we get to the termination sequence or a stop codon. Stop codon initiates release, and the ribosomal subunits dissociate. I talked more about RNAi in class, but there's a couple links to some videos to watch about that. Because once the protein is released, that RNA gets chopped up. That's one of RNAi's jobs, is to chop up any mRNA that needs to be recycled. There is also the idea that there are a lot of ribosomes that can work on the same mRNA strand at the same time. So you can make a lot of proteins or a lot of amino acid sequences or chains all using the same mRNA transcript. So that's what's being shown in that picture. Then that amino acid chain needs to be folded and made into something functional. There are these proteins that exist. They are embedded in the ER or in other membranes that are like vesicles. The Golgi has these proteins embedded within, and it provides like a buffer for these amino acids to fold without being influenced by molecules nearby. Because remember, it's all about those R groups that have to interact with one another to do proper folding. And if there's other charged particles around, even water can influence how well it folds. So it creates a nice little hydrophilic environment so that it doesn't get disrupted by all those other intermolecular forces. Don't forget, too, that there are a couple different types of ribosomes. The location is going to determine its function. So the free-floating ribosomes are going to be making proteins that stay in the cell and function maybe in signaling cascades or in some kind of amplification or metabolism of something, but basically as part of an enzyme for the cell's function. Then you have bound ribosomes, and those ribosomes are busy making proteins for export, ones that need to leave the cell as a cell product. And don't forget about our endomembrane system, starting there at the nuclear envelope, working its way through the ER, shipped to the Golgi, packaged into vacuoles or vesicles, and then headed for the outer membrane for secretion. Now, how does it know where to go? Of course, chemistry. There's a signal peptide or a type of protein that acts kind of like a cellular zip code, right? It's going to tell the protein where it's supposed to end up, whether it's at the outer membrane for excretion or if it's supposed to go to a free ribosome and stay there in the cell. So these are, again, signaling peptides. And these are called SRPs. You can see in the picture there that it's orange, these signal recognition particles, and they cleave off that green part, remove the signal peptide when a protein is ready to be packaged or shipped. Mutations is the last part of chapter 17. Don't forget that the DNA level is where the mutation occurs. So if the mutation happens after that, then it really only affects one protein in hundreds, and that mutation is doesn't even count, right? It doesn't ever get seen. If the mutation is in DNA, then every protein that ever gets made using that gene will be incorrect. And that then will have an effect on the final protein. So when we study mutations, we work at the gene level, the DNA level. A large scale mutation is called a chromosomal mutation. And that is when an entire piece of chromosome containing tens of hundreds of genes can break off or invert itself. Those always cause major challenges and difficulties for the cell and for the organism, maybe even death. This can be caused by non-disjunction during meiosis, meaning that when the homologs are supposed to separate in anaphase 2, they don't. They accidentally stick together and end up in the same gamete. So you could end up with total chromosomes missing or an extra chromosome. Down syndrome is a result of non-disjunction of chromosome number 21. Translocation means a piece of a chromosome breaks off and actually adds to another one. So the genes aren't lost, but they are now added to a different chromosome, which means they won't be read or controlled the same way. Inversions literally means that the chromosome breaks off and then reattaches in the opposite direction, upside down. All those genes will be read in the wrong order and therefore will not code for the proper proteins. Duplications, pieces of a chromosome get copied over and over, repeated. And then we have, of course, large deletions, which is exactly as it sounds. A huge piece is deleted or eliminated. Point mutations are the ones that we can track in proteins, however. One base pair that is off can change the final protein sequence. And there's different ways that this can happen. One of them is called a base pair substitution, which is when a wrong nucleotide gets stuck in there with another one. So if a C was supposed to go there, but a U is accidentally put in, or a G gets put in. That means that one amino acid or one codon could be changed. Now, it may not be changed depending on which nucleotide in the triplet is affected. So if one of the codons gets changed and now codes for a different amino acid, that is called a missense mutation. If it accidentally codes for a premature stop codon, that's called a nonsense mutation. Nonsense mutations are not amino acids anymore. So it codes for a stop codon, not an amino acid, which means all the other amino acids after that are not going to get put in place. And that will be a malfunctioning overall amino acid chain. A missense sometimes goes unnoticed because just one amino acid different could change the way it folds, as in the case of sickle cell anemia, or maybe not. Maybe it doesn't change it and it can still fold properly and work pretty well. So those don't always affect the final protein. Is there silent substitutions? Is there a way that you can make a mistake that goes completely unnoticed? Sure there is, right? Remember wobble? That third base in a codon triplet, a nucleotide triplet, doesn't really matter in most cases. So if the mistake is in that third base, everything will happen just as it's supposed to. Everything will be fine and the protein works. And that is what's known as a silent mutation. Then we have frame shifts. Frame shifts are always bad. Basically, the nucleotide gets added or taken away, so not just substituted, but you end up with an extra one, which shifts the entire reading frame. Every single codon after the nucleotide is either deleted or added is going to be messed up. It always results in a non-functional protein. Again, the only way to shift your frame is to either take away a nucleotide or add an extra one, and that will shift your reading frame down. So this is just a picture that goes with the words from the previous slide. A missense mutation, again, is just one amino acid that's different. So in this case, glycine was swapped for a serine. A nonsense mutation, in this case, we had the lysine accidentally got switched to a stop codon. So it stopped as soon as it started. This will never exist as a protein. And then a silent, if you look at the amino acid methionine, lysine, phenylalanine, and glycine, then look down there and it says the same thing. We changed an A to instead of a G, and now it ended up not mattering at all because it was the third base. Frame shifts, again, adding something. So notice the picture at the bottom, we have a little bit of a longer strand because we added an extra A, and that corresponded to an extra U when it came down to reading, which accidentally coded for a stop or any other sequence of amino acids. Either way, it's not going to be the right order. Deletion, same thing. You're going to mess them all up. We're going to remove an A somewhere downstream. We start with methionine. We get our lysine in place, but instead of phenylalanine, we bring in a leucine, and everything else is messed up. Again, this is sickle cell as an example of a condition created by just one nucleotide mutation. It's actually just one amino acid, so that's different. Instead of a glutamate, it's a valine, and this is what causes the misshapen folded hemoglobin, and therefore it cannot carry oxygen as effectively. It forms long, inflexible chains, which causes the red blood cell to have that weird sickled shape instead of a nice, flexible donut shape, which means they're much more likely to get caught and stuck on each other and create clots in blood vessels. Again, the last part of this chapter was just to reiterate the difference between prokaryotic and eukaryotic protein synthesis. Prokaryotic, the DNA is read and directly translated into mRNA that is used directly to make a protein. There is no mRNA processing, so there is no pre-mRNA made. Everything happens in the same part of the cell. There is no nucleus. And then in eukaryotic cells, we have those extra steps. So this is it in writing instead of in a picture. So it happens in the cytoplasm versus separation of steps, nucleus then cytoplasm. Again, DNA and RNA are all in the same location versus RNA traveling to the cytoplasm out of the nucleus. RNA polymerase binds directly at the promoter in prokaryotes, whereas in eukaryotes, we have an extra level of control. RNA polymerase binds to the TATA box, and transcription factors have to join up in order for transcription to begin. So again, that gives us just greater levels of control. If the TATA box is mutated or something, then that will affect gene expression. But we also have a way to turn on and off genes and keep them from being expressed if we don't want them. We can put things to block transcription factors and keep them from getting access to the DNA, and that will keep the gene from getting read. Again, transcription makes mRNA directly, no processing, whereas in eukaryotes, we have an option to make different mRNA templates from pre-mRNA because we have RNA processing and alternate splicing. And that's the last little part, the no splicing. In prokaryotes, there is no interons and exons. We don't remove any portion of it. It gets read directly and used directly. Here's a beautiful picture that summarizes everything that we talked about. And I hope that makes things more clear for you and helps you feel comfortable and confident and prepared. Okay, now I am going to stop the show if I know how to do that.